Hi, everybody. My name is Ed Davis. I'm Senior Application Scientist at GeneCopoeia Incorporated, located in Rockville, Maryland, just outside of Washington, D.C. I'd like to thank you all for joining us for today's webinar entitled Antigen Protein Microarrays for Effective Antibody Profiling. We really appreciate it. Before I get started on the webinar, I'd like to go over a few practical matters. First, you may ask me questions at any time. To do so, please type your questions in either the Q&A function or the chat function. Secondly, in the interest of keeping the talk flowing smoothly, I will wait until the end of the webinar to answer your questions in the order in which they were received. In addition, I will read each question out loud and provide the answer out loud for everyone to hear. Okay, so with that, I'd like to get started. First, I'd like to tell you who we are at GeneCopoeia and what we're about. We were founded over 20 years ago as a global provider of plasmid clones, specifically originally ORF clones, that's open reading frame clones, which allow you to express virtually any mammalian gene, primarily for cell lines, but they can be used in other models too, such as transgenic mice, etc. Since then, we've expanded these offerings to promoter clones, that is gene promoters, microRNA clones, that could be microRNA precursors, inhibitors, or 3' UTRs, which are targets for microRNAs. We have a number of CRISPR solutions, including plasmid clones, as well as other reagents for gene modification. And finally, we have shRNA clones for gene silencing. Next, we have viral systems. That is, we provide clones and viral particles for lentivirus, AAV, which is known as adeno-associated virus, as well as adenovirus, a more traditional DNA virus. Further, we have a number of solutions for stable cell lines, including CRISPR-Cas9 stable cell lines, which is the topic of another webinar, labeled cancer cell lines, which are used for in vivo tracking of tumors and their responses to various therapies. And we also have cancer biomarker mutant cell lines, which are mutant cell lines containing mutations in a number of cancer hotspots. We also have stable cell line services, not shown here, for construction of virtually any cell line of your choice using various methods like CRISPR or ORF overexpression. And finally, we have a number of solutions for kits and reagents, including transfection, luciferase, FISH probes, fluorescence in situ hybridization probes, indel detection, which is a CRISPR-related kit, cloning kits for building your own plasmids, and finally, what you came to hear me talk about today, antigen microarrays. GeneCopoeia has a number of solutions for antigen microarrays under the brand name OmicsArray. First, we have 7 catalog pre-made arrays, and I'll tell you more about the structural detail in a moment. But these are pre-made protein microarrays that are geared towards specific applications or disease or studies. The most popular antigen microarray we have is the Human Autoantigens General Survey microarray. And I'll go over these in some detail in a little while, but we also have an array focused on brain and central nervous system disorders, cancer and neoplasms, common allergens, and then we have a number of newer arrays for autoimmunity and their relation to pathogens, such as this human autoimmunity allergy and infection array, a human coronavirus associated autoimmunity array, and finally, a brand new array called SARS-CoV-2 coronavirus variant proteins. But that's not all we have. custom arrays are also available. So if you wanted to have us create an array with proteins or antigens of your choice, we can do that. The pricing depends on several factors, such as the source of proteins and the number of proteins. So that would be considered a custom project. Okay, so what are OmicsArrays antigen microarrays? Well, for one thing, these are arrays in which proteins or other molecules of interest, they're almost always proteins, but they can be other macromolecules, like double-stranded DNA, for example, in which they're spotted on a nitrocellulose membrane-coated glass slides. And these are intended to capture antibodies from patient samples. Now, the nitrocellulose membrane is important because it's a similar type of membrane that's used for traditional Western blotting. Okay, and it provides very high binding capacity, higher than some of the synthetic polymers that other companies use for similar types of protein microarrays. The antigens are selected based on their association with specific diseases, biological functions, or pathogens. Antigen microarrays are used for detection of antibodies from various sample types. That's why they're antigen microarrays. They're a special type of protein microarray. So they're meant to capture antibodies. And these various sample types can include serum, plasma, saliva, other body fluids, or cell culture supernatant. They can also be used to validate specific antibodies, like monoclonal antibodies, for example. The antigens are usually known autoantigens in autoimmune diseases, but they can be other proteins associated with other disorders too, such as infectious diseases like COVID-19. And antigen microarrays can also be used to monitor the antibody response, that is the humoral immune response to vaccines. So these arrays that we have are, as I mentioned, they're nitrocellulose coated glass slides. The way this works is we have glass slides that actually contain 16 squares or pads made out of nitrocellulose that are adhered to these glass slides. Each of these 16 arrays are duplicated, are replicates. Okay, so we spot up to 256 spots of antigens on each array. And because we typically use up to 120 antigens plus 8 controls, that means each spot is duplicated on the array. So we'll make 256 spots on each of 16 pads. There are 16 identical arrays on one slide. And what this allows us to do is it allows us to probe or assay 16 different samples on one slide. We usually reserve one as a negative control. But then that means that we can probe up to 15 samples on one slide. So this can be like, for example, 15 different patients that are showing autoimmune symptoms, for example, or 16 different monoclonal antibodies. So it's a relatively high throughput format compared with some other companies' microarray formats. So what are the types of antigens that we put on these OmicsArray antigen microarrays? Well, as I mentioned, they're primarily proteins, but not always. So it can be proteins, nucleic acids, drug probes, enzymes, which are of course proteins, but they're specialized. We typically use whole proteins or full-length proteins, I should say, but we can also spot peptides. We can also spot whole pathogens as well as allergens. Okay, so there can be several different types of macromolecules. The OmicsArray product line has several advantages over various methods and other array types. Number one is multiplexing. So the OmicsArray antigen microarrays, as I mentioned before, can assay up to 120 antigens at a time. That's compared to only one protein at a time for ELISA, which is a more traditional method that you're probably familiar with. We also, as I mentioned, we typically use full-length proteins. Most of the proteins are full-length, which provides more opportunities for antibodies to recognize natural epitopes. So that using a full-length protein is more likely to give you the normal three-dimensional structure of a protein. Now, you can use peptides, as I mentioned before, and that can provide certain advantages for like epitope mapping, for example, and we can do that. But often, as you know, the peptides are not presented in native structure, so they may not, they could provide false negatives. They may not bind a naturally occurring antibody. Further, as I mentioned before, these arrays provide a relatively high throughput format. So each slide can process up to 15 samples in parallel. So 10 slides can be 150 patients, for example, or 150 antibodies. This allows for high flexibility. This allows for a combination of different antigens, such as proteins, peptides, nucleic acids, etc., as well as detection of different antibodies types and subtypes like IgG, IgA, IgM, etc. OmicsArray antigen microarrays also provide high sensitivity. So each array can detect as little as 1 picogram per mil of antibody, which is 100-fold more sensitive than ELISA. And finally, they also provide they're also capable of allowing for small sample volumes. So as little as 1 microliter of serum is needed for detection. This slide shows the antigen microarray workflow. So we start out by doing robotic printing of the antigens on the array, and we use inkjet printing to print our proteins and other antigens on the array, and that's more accurate and consistent than more traditional pin spotting. And so we create the arrays by robotic printing, and then we do a hybridization step with the patient samples or with the monoclonal antibody, whatever the sample is. And typically, what we will then do is we will hybridize or incubate with the sample, and then we'll wash it and then probe with a fluorescent labeled secondary antibody. Typically, we probe with 2 different labeled secondary antibodies. What people most often go for is either labeled anti-IgG and anti-IgM. We will typically label the IgG with Psi3, which for us is in the approximately green wavelength, and we'll label IgM with Psi5, which is a far red fluorophore. And so then when we wash off the excess secondary antibody, we'll then simply put the slides into a microarray scanner, a very standard microarray scanner, very similar to DNA microarrays. And then we get a readout. This shows just the Psi3 channel because it's green, but we would get a similar readout with the Psi5. And then we subject the fluorescence intensity is subject to data analysis, usually by heat mapping. So we can provide a heat map which shows the signal intensities where increasing red on the heat map means a greater signal intensity. The autoantigen arrays do undergo quality control procedures. So we have internal controls included in each array where spot variation CV is less than 10%. Okay, so what are some of the applications for antigen microarrays? Well, there are many. Shown here are just a few. So one of the most widely used or pursued applications is autoimmune disease profiling and diagnostics. So there are many autoimmune diseases such as lupus, which is also known as systemic lupus erythematosus, rheumatoid arthritis, Crohn's disease, type 1 diabetes, Sjögren's syndrome, and there are up to 100 other known, well-characterized autoimmune diseases. Another application for antigen microarrays is autoantibody profiling in response to cancer immunotherapy. So, as you may know or may not know, one of the prominent therapies for cancer treatment is immune checkpoint inhibition, most widely through the PD-1, PD-L1 pathway. These are very powerful anti-cancer therapies, but one potential drawback is that, especially with PD-1, PD-L1, anti-PD-1, PD-L1 therapy, is that you can trigger an autoimmune response, because the natural function of PD-1, PD-L1 is to put the brakes on the immune system to prevent autoimmunity. So one thing this can be done and has been done is that these arrays can be used to monitor patients while they are undergoing immune checkpoint inhibition therapy. Another application is evaluating the response to infection by pathogens such as SARS-CoV-2, the virus that causes the COVID-19 pandemic, as well as respiratory syncytial virus, influenza, etc., or in response to vaccination against these agents. Another application is autoantibody biomarker profiling for neurological and neurodegenerative diseases. As I mentioned, we have an array based on that. There's allergen profiling. So instead of having a patient undergoing traditional subcutaneous injection of an allergen, which can be very painful and uncomfortable and inaccurate, we can instead create an array with different allergens spotted on the membranes and then probe patient samples for antibodies against these allergens. And finally, but not last but not least, is evaluation of the immune response to organ transplantation. So again, these arrays have been used for a number of times in various applications, such as, for example, autoantibody profiling in lupus. Our arrays, the developer of these arrays has had a number of publications going back several years for lupus, as well as immune mechanism studies, or even other immune-related diseases such as autoimmune muscle disease, rhinosinusitis with nasal polyps, and discoid and systemic lupus patients. Or obesity is another example, Sjogren's syndrome. Or COPD, chronic obstructive pulmonary disease. I also mentioned organ transplantation. These arrays have been used for studying the reaction to organ transplantation. And cancer immunotherapy. There are a number of papers for these arrays, as I mentioned, that have assayed patients undergoing cancer immunotherapy. Further, another application, which I did not even mention, but that's to evaluate the effect of nutrition supplements on environment-induced autoimmunity. We have a couple of papers on that, as well as pathogen and vaccine studies, such as dengue and influenza, and everybody's favorite pandemic, COVID-19. We have some array studies, papers that have used our arrays for that. Okay, so as I mentioned before, we have a number of pre-made arrays, 7 to be exact, that I've shown here. So I'm going to go over these a little bit in detail. I'm going to start with the Human Autoantigens General Survey Array, which is our most popular array. So this has a number of important features. Again, like most of our arrays, not all, but most, this profiles or contains 120 antigens plus 8 controls, and these are IgG controls. Antigens known to be associated with specific autoimmune diseases are chosen based on a thorough review of peer-reviewed publications. And some of the examples of the autoimmune diseases represented in this array, as I mentioned before, rheumatoid arthritis, muscular dystrophy, lupus, type 1 diabetes, and many others. This is a partial listing of the antigens on the array, exactly half to be exact, just to give you an idea of what we have on this array. We do not show the entire list on our website, but the array lists, the antigen lists are available upon request. Now I'm going to talk a little bit about this array, the human brain and central nervous system disorders array. So again, this features 120 antigens plus 8 controls, and it profiles autoantibodies associated with known neurological disorders. These antibodies could be either causes of disease or biomarkers associated with disease. And again, this is a partial listing of the antigens, 60 of the 120. And as you can see, there are many that are associated with specific neurological diseases like Alzheimer's. Next, I want to talk about the human cancer neoplasms array that I mentioned before. It can be used to profile patients' autoantibody response for immune checkpoint inhibition. This also contains 120 antigens plus 8 controls, and it profiles autoantibodies associated with response to cancer immunotherapy, which can induce autoimmunity in some cases. And this is also a list of 60 of the 120 antigens on the array. And the complete list is available upon request. And finally, the last array I wanted to talk about in some detail is our new SARS-CoV-2 coronavirus variant proteins array. This profiles 58 antigens plus 8 controls. It contains proteins from SARS-CoV-2, other coronaviruses, such as the original SARS virus and MERS, influenza virus, respiratory syncytial virus, etc., are included on the array. And it also, so far, this is the only one in the market, but it also includes SARS-CoV-2 variants. So it contains spike protein, partial spike, not full length, partial length spike proteins from some of the various variants, such as the alpha and delta variants, as well as some others. And because there are only 58, I was able to put the complete list on this slide. This complete list is also available on our website. And as you can see, it contains not just SARS-CoV-2 spike protein, but also other proteins from SARS-CoV-2, such as some of the non-structural proteins like ERF3A, 7A, etc., as well as MERS and the original SARS-1, and some other pathogens such as adenovirus, respiratory syncytial virus, and Coxsackie virus. Coxsackie virus is important, incidentally, because it may be a risk factor. Exposure to Coxsackie virus may be a risk factor for type 1 diabetes in children. And as I mentioned, this array contains some of the known variants. This spike protein shown here is from the alpha variant. This one is from the beta variant, it's also known as the B.1.351 variant. This is from the gamma variant, which is also the P.1 variant, originally identified in Brazil. And of course, we have spike protein from S1 domain from the Delta variant, which is now the predominant SARS-CoV-2 variant in the world. Now I want to show you a little bit of the performance data from the SARS-CoV-2 coronavirus variant microarray. This is an internal study that was done, and this shows groups of patients. So we have 3 groups of patients, unvaccinated, vaccinated with one of the approved the US approved SARS-CoV-2 vaccines, as well as COVID-19 patients or convalescent patients. So what we did here was we took the serum from these various volunteers and patients, and we probed our coronavirus variant antigen microarray with these sera. And this, what we see here is the net signal intensity, the normalized signal intensity, average normalized signal intensity of each group to different antigens along the array. Each of these groups of bar graphs is a specific antigen on the array. So there is some response to some pathogens, especially the common cold coronaviruses such as HCOV-229E, for example, as you would expect. For some of the non-spike proteins, some of the non-structural proteins for SARS-CoV-2, such as the nucleocapsid protein, we only see a response in convalescent serum. And this is not a surprise because, as you probably know, none of the US approved vaccines from Pfizer, Moderna, or Johnson & Johnson use any of the non-spike proteins as an antigen. They only use spike protein. So only a natural infection will give you a response to these other proteins. Same is true of ORF3A, for example. Now, when we look at the antigens that are derived from spike, from the wild type and the different variants, we see a different story. So unvaccinated, typically we see, that's the blue bar, we typically see little to no response. But then upon vaccination or in response to infection, we can then see a fairly robust response. And again, these are the average net signal intensities among each group. The next study I wanted to show data from for the SARS-CoV-2 variant antigen microarray is where we use this array to follow the durability of the immune response over time. So in other words, to look for waning antibody response, which of course is a topic of high relevance currently as it pertains to booster shots, as well as new vaccine development. So what we have here now, what we're looking at here now is a similar type of experiment where we have different groups. In this case, it's now 5 different groups. Unvaccinated, shown by dark blue. 2 weeks post-vaccination, shown by the orange. 2 months post-vaccination, shown in gray. 4 months post-vaccinated, shown in yellow, and 6 months post-vaccinated, shown in light blue. Now we're only looking at the SARS-CoV-2 spike antigens on the array. And again, these are the average net signal intensities looking at the IgG response in these different groups. So as we would expect, in the unvaccinated group, we see little to no signal, okay? And then we get a robust response 2 weeks post-vaccination to most of the antigens. And then what we see is we do, as we expect, we see a little bit of a waning over time. So there's not much difference between 2 months and 4 months post-vaccination. It is lower than 2 weeks post-vaccination. But then by 6 months post-vaccination, we do see a significant drop. And again, remember, this is done in parallel, looking at many patients in parallel, 15 patients per slide. Okay, and then looking at many different antigens on the array. Okay, now I want to shift gears a little bit and talk about custom antigen microarray services. So I did mention that we have the pre-made arrays available or custom arrays available. You can purchase the slides, and then if you want, you could be on your own and do all of the work yourself. But of course, we offer custom services as well. For one thing, we have custom printing and spotting, where we will create custom antigen microarrays built to your specifications. We also offer sample processing, so customers can send blood, plasma, tissue, or any other biological sample, and we'll prepare the samples for processing and incubation with any of our pre-designed antigen microarrays or custom-built antigen microarrays. So we offer basically a full profiling service. So You basically can just send us your samples and we'll do all the work and we will send you a report, which includes the data analysis as well. So once samples are processed and incubated with an antigen microarray, we will analyze the raw data, and the standard analysis service includes an Excel file of the net signal intensity for each antigen on the array, normalized to internal controls, as well as a heat map. Additional analysis services, including proteomic analysis, pathway analysis, and more, are also available. This is an example of the data that will send to the customers. So this is the final data output format. It's an Excel file. I know this is a little hard to read, and I apologize for that. But what we have is along the top row, we have each sample. So each patient sample, for example. And then in each row, so that's in each column is each patient sample. And then each row represents an antigen on the array. So what you get is the signal intensity, the net signal intensity for that patient sample for that specific antigen, all in an Excel file. We also provide a heat map for each antigen on the array for each individual patient sample. And again, the redder it gets, the higher the signal intensity it gets. So that was the standard data output that we provide, but we also can provide additional analysis such as gene ontology or GO analysis, which classifies genes and proteins based on known biological functions. So we can tell you, hey, you know, these particular hits that you got are enriched in these biological pathways or biological functions. The same is true of pathway analysis. That's also a known analysis service that we can do, which also groups positive antibody-antigen interactions based on defined biological pathways. Okay, so with that, I would like to summarize. First, I told you that GeneCopoeia's OmicsArray antigen microarrays are designed for multiplex high-throughput profiling of antibodies in many different body fluid types, such as whole blood, serum, plasma, saliva, etc. GeneCopoeia's OmicsArray antigen microarrays are a great solution for many applications, including autoimmune disease screening of patients, cancer and neurological disease autoantibody profiling, and detection of pathogens, including SARS-CoV-2, the virus responsible for COVID-19. And finally, I told you that GeneCopoeia offers several high-performance pre-made antigen microarrays, as well as full custom antibody profiling services. Custom arrays are also available. Okay, so with that, I would like to stop and take your questions. We do have a number of questions, so as I mentioned, I will read each one out loud and then read the answer out loud for everyone to hear. Okay, so the first question, and I'm not going to say who sent these questions, I'm just going to read them. Are the proteins made in mammalian cells? That's a really good question. The answer is fairly complicated. In some cases, they are made in mammalian cells, but not always. We use proteins made in mammalian cells, bacteria, cell-free systems like wheat germ in vitro translation systems, baculovirus systems. So essentially, we don't necessarily pick the method. It depends on what is available at the time. So a lot of the proteins we use are actually commercially available proteins. Not always. Some are produced in-house, and when we produce them in-house, we will try different methods. It would be nice to use bacterial methods because that's convenient, but of course the proteins don't always fold correctly or get correct post-translational modifications, for example. So again, it depends on what's available. Okay, so next question. Can you make chips with more than 120 proteins? Yeah, the answer is yes, we can, but we cannot at the time. We cannot currently fit more than that on the standard 2 by 16 array format. So in other words, the chips on the 2 by 8, the chips on the 2 by 8 formats are only big enough to hold 256 spots. And we, as I mentioned, we spot each protein or antigen in duplicate. So we could increase that number, but then we would have to go to a smaller format, which we can do. We can produce slides with 4 arrays, for example, instead of 16. So we can do that. The downside is that you would not be able to probe as many samples at one time if you want. Okay, so next question. Do you have antibody arrays? Unfortunately, yeah, that's a good question. At this time, we don't. At this time, we don't have antibody arrays. We are considering adding those to our repertoire in the future, but not at the moment. Okay, so next question. Can I get a recording of the talk? Yes, that's a common question. The answer is yes. This talk is being recorded, and so we will post it on the website within the next 24 hours. And everyone who registered for this webinar will get a link to the recording. Okay, next question. Are the arrays validated? Yes, that's a very good question. We do some validation of the pre-made arrays. What we do is for some of the arrays, we will run Western blots for some of the antigens and then probe them with a commercially available antibody. So that's the extent of the validation. Okay, so the next question is, how do you label the samples? Okay, well, we don't really label, if you're talking about like fluorescent label, we don't label the actual patient samples. Well, as I mentioned before, we label secondary antibodies. So the patient samples, the antibodies in the patient samples will bind to antigens on the array, and then excess antibody is washed off. And then we probe with anti-IgG or anti-IgM or anti-IgA, and those anti-IgG antibodies, those secondary antibodies, those are fluorescently labeled with either Psi3 or Psi5. And then we can detect them on a microarray scanner using the proper filtration. Okay, so does anyone have any other questions? Okay, well, if not, again, I would really like to thank you very much for attending this webinar. Again, a recording will be available within the next 24 hours, and you will all get an email with that link. So if you have any further questions that I've not been able to answer today, feel free to call us at 301-762-0888, excuse me, or send an email to inquiry@genecopoeia.com. All right, with that, I'd like to stop, and everybody have a good day. Bye.